Okay, so welcome back from your break, whether you took a full break or whether you just clicked to rejoin this section. Welcome back. Let's take a look at the concept check question, and then we'll move on and talk about mitosis in more detail. So eukaryotic organisms use two types of cell division. Remember, we're talking about mitosis and meiosis. What are the functions or roles of these two cell divisions in the life of a typical eukaryote? So this was basically what we were talking about right at the very end of the last section. So mitosis, remember, is for growth and development. We all develop from the single cell that forms from the fusion of the egg and the sperm, the zygote. So all of our cells are the product of mitosis, so growth and development. Then repair and replacement. So when tissue gets damaged, skin gets cut, those sorts of things, we have to do repair, and that involves cell division in the form of mitosis, and replacement. When we turn over our skin, when we turn over our red blood cells, we're doing replacement. And again, that's mitosis, where we're creating identical copies of the cells that we are replacing. Meiosis then is involved in sexual reproduction for a typical eukaryote. Even those that may depend on asexual reproduction primarily, a lot of eukaryotes that do asexual reproduction still feature sexual reproduction because it creates genetic diversity. So for a typical eukaryote, meiosis is involved in sexual reproduction. That's its role. And it produces the gametes, the eggs and the sperm, the reproductive cells. Okay, so let's move on and start talking about mitosis, because that's the topic of this next section. So the first thing to remember is that for a eukaryote, the genetic material is the DNA, and that DNA is in the form of linear chromosomes bundled up inside the cell nucleus. We do know that there's DNA in mitochondria and chloroplasts. It's in the form of small circular molecules, because remember, mitochondria and chloroplasts are derived through endosymbiosis from prokaryotes, from bacteria in the past. If that sounds a little bit fuzzy, you might want to go back and take a look at the chapter where we looked at organelles, which, if I remember rightly, is chapter 5. Because we talked a little bit about the endosymbiosis theory of the origin of mitochondria and chloroplasts there. So there is DNA with genes outside of the nucleus, but predominantly the nucleus is the location of the DNA in a eukaryotic organism. And so those chromosomes are the source of the genes that need to be passed on from one cell to the next whenever a cell replicates itself. So whenever cell division occurs, those chromosomes are what are going to be passed from one generation of cells to the next generation of cells, or big picture, from one generation of organisms to the next generation of organisms. In fact, the chromosomes and the genes that they contain are the only things that pass from one generation of cells to the next, or from one generation of organisms to the next. And so there are some interesting ideas that biologists have developed out of that simple observation that the chromosomes are the only things that move from one cell generation to the next generation, or from one generation of organisms to the next generation of organisms. Unfortunately, we don't have a lot of time to talk about that. So unfortunately, all I can do is tease you with that. But there are some interesting ideas that come out of that. Basically, it boils down to the fact that organisms are genes' way of making copies of themselves. That would be a good summary of it. So an acorn is a wave, or sorry, an oak tree is an acorn's way of making more copies of itself, or a body is the genes in the nucleus's way of making copies of themselves. That's basically what it summarizes as. That the only thing that really matters long term is the survival of the genes, not survival of the organisms. Because the only thing that moves from one generation to the next are the chromosomes containing the genes. So the only thing that really matters is the survival of the genes, not survival of the organisms. So the genes make sure that they survive by making copies of themselves through the organisms that they produce. So organisms are considered to be the gene's way of making copies of themselves. That's the notion. Unfortunately, we can't delve deeper into that, but it is an interesting idea. It's called the selfish gene concept, if you're interested, and you can look that up and read more about it if you want. So one of the things that then becomes really important is that if this is the thing that's going to be passed from one generation of cells to the next generation of cells, or one generation of organisms to the next generation of organisms, prior to any cell division, we need to make a copy of those chromosomes. So that's implicit in the idea of cell division. At some point prior to cell division, there needs to be a copying of the chromosomes. And the number of chromosomes that are going to be present has nothing to do with the complexity of the organism, which is illustrated in the next diagram. So if you look through this diagram, you can see that every species listed here has a different number of different number of genes, or sorry, chromosomes. But the number of chromosomes is not a reflection of just how complicated the organism might be. So the organism with the greatest number of chromosomes is one that we might consider to be rather simple compared to ourselves. It's a type of rat. Actually, it's a relation of the gerbils and the guinea pigs, lives in South America. And a deer, for all the difference in size between the deer and this rat, the deer have a limited number of chromosomes compared to the rat. Now, we could add in here, and this is something that is not listed here, Drosophila. Oops, get the pen going again. So Drosophila, or the fruit fly, because this is an animal that we will be talking about in the next chapter. We'll talk a little bit about what we've learned from Drosophila in terms of genetics patterns, because we've learned a lot. Drosophila have 8 chromosomes. And why that is interesting is that we have often compared ourselves to Drosophila in terms of the number of genes that are required to make each type of organism. So Drosophila, it turns out, have about 17,000 genes. Now, some people say as little as 15,000, but the number is between 15 and 17,000. Humans have somewhere between 21 and 23,000. And I don't think anyone would argue that a human is far more complicated than a Drosophila. And yet the difference between the number of genes is rather small, comparatively speaking. It's all about what you do with those genes. Now, you notice that humans have 46 chromosomes, and Drosophila only have 8. So what that tells you is that the density of genes on the Drosophila chromosomes is very high compared to the density of genes on human chromosomes. So complexity, number of chromosomes, density of genes, number of genes has nothing to do with the complexity of the organism. It's just what is. So some organisms have hundreds of chromosomes, but compared by our standard at least, we would consider them to be fairly simple. Whereas other organisms that are quite complicated have a limited number of chromosomes and a limited number of genes, comparatively speaking. There is no set pattern that you can observe in terms of complexity of organism, numbers of genes, and numbers of chromosomes. It's just what is. So that's always something to bear in mind as we're talking about this. But the point behind this is those chromosomes, remember again, are what are going to be passed on from one generation to the next. So one of the issues that we have with all of this genetic material is packaging it into the nucleus. The nucleus is quite a small organelle, and quite a small space. If we were to think in terms of the chromosomes that we possess, if we took all 46 of those chromosomes and linked them together in a long chain, they would stretch for roughly 10 feet. Yet they've got to be packaged into a nucleus whose diameter is measured in millionths of a meter. What is referred to as a nanometer. Sorry, a nanometer is 10 to the minus 9, so that would be 1,000th of a millionth of a meter. Very, very small unit of length. The point is, we would have a real issue if that DNA was a single long linear molecule. We'd have a tough time fitting it in. Fortunately, it's not. The first thing is it consists of 46 separate molecules, the 46 chromosomes. Then rather than being long, thin, stretched out molecules, it's actually complexed with proteins called histones that creates a molecule or a form that we call chromatin, and you can see the name here. So this is the normal form in the nucleus. And it's not visible. So when you put a typical eukaryotic cell under the microscope, and even with a really good light microscope that allows you to see some detail of the nucleus, you're not going to be able to see the chromosomes. They're simply invisible when they're in this chromatin form. The only time that you can actually see the chromosomes through the light microscope is when the cell is in the process of dividing. And in fact, this is one of the clues that lets us know that a cell is beginning to start the division process, because you can actually see the chromosomes in the nucleus. It actually goes through a process which we call condensation. We say that the chromosomes condense. It shortens them and thickens them in such a way that they become visible, and they're coated with a layer of protein that increases the thickness of the structure so that they become really visible. So this is basically what it looks like. Here you can see the chromosomes in this cell. It's in the process of dividing. And so you can see that the chromosomes are quite thick, comparatively speaking. Next door is a cell that is in a non-dividing stage of this, what is referred to as the cell cycle. This stage is called interphase. And you can't see the chromosomes at all. They're just simply invisible. They're there in their chromatin form. They're just not visible. But once the cell starts dividing and the chromosomes condense and get complex with protein, then they start to become visible. So this is how it takes place. So here is the quote unquote naked DNA, the famous double helix. It gets complex with these special proteins called histones that are only found in the nucleus of eukaryotic cells. And you can see that the DNA wraps itself around the histones. And this is what gives rise to the chromatin. So this, oops, I was trying to write on the wrong screen. I do that occasionally. So this is chromatin. So this is what you would expect to find if you could see the DNA in a microscope, which you can do with an electron microscope. So this image over here is actually an electron microscope image of chromatin. You can see the histone beads that the DNA is wrapping around in the picture there. But that's with an electron microscope, and that's magnified somewhere around 20 to 30,000 times. A good light microscope struggles to magnify around 2,500 times. So you can see the difference. Then when the chromosome condenses, it goes through two steps. So it has this step, This is called coiling. And it's pretty obvious why it's called coiling, because it wraps itself up in a coil. And then it layers itself this way, and this is called super coiling. And this is these two together, coiling and supercoiling. Actually, I won't say at, I'll say in. So what we've got here are the condensed chromosomes. And in fact, these are how you would see the chromosomes as they condensed and became visible at the beginning of cell division. So the chromosomes have already been duplicated. I've mentioned previously that prior to cell division, the chromosomes need to be copied. So this is a duplicated chromosome or a copied chromosome. The two copies are sitting here. There's, turn that on. So here's one copy, and here's the other copy. And these are referred to as sister chromatids within the chromosome. So the unfortunate part about scientists is that they sometimes tend to use the same word to describe different things. And we use the term chromosome very loosely at times to describe several different structures that can be observed in cells. So the chromatin form of the DNA, invisible as it is still referred to as a chromosome. When the chromosomes are copied, each of the copies then exists within a chromosome, but you can't use the name chromosome for each of the two copies, so we refer to them as chromatids. So each copy of the original chromosome is now referred to as a chromatid. They're called sister chromatids because they're copies of the original DNA molecule. And so when the chromosomes condense and become visible during cell division, initially you see the two sister chromatids within the same chromosome sitting next door to each other, and they're held together by this complex called the centromere. And during the initial part of both types of cell division that we're talking about this morning, it's really important that the chromatids stay together and be moved around together. Eventually, they will be separated because each one of these sister chromatids will eventually become an independent chromosome. So eventually they will separate. But during the initial part of the division process, they need to be anchored together so that they can be moved around together. So why condense the chromosomes? Well, for two reasons. Number one, it's easier to move them around in this condensed form than it would be if they were in their chromatin form. It's, just think about it this way, which is easier to move, a 10-foot length of string or that same 10-foot length of string rolled up into a little ball. And obviously the answer is the rolled up little ball. A lot easier to move it around. So by condensing the DNA molecules down to these tight packages, it makes them a lot easier to move. The other reason for doing this is that once you condense them down and coat them with the protective protein, you now protect them from the cytoplasm, because during cell division, the nucleus, the nuclear envelope, actually does break down And for a short period of time, the chromosomes are actually exposed to the cytoplasm. And it turns out that the cytoplasm is a really hostile place for chromosomes, which is the reason why they're usually locked away inside the nucleus behind the nuclear envelope. But for a short period of time during cell division, they're actually exposed to the cytoplasm. So to protect them, because they need protecting, they are the genetic material of the cell, you condense them down and coat them with a protective coat of protein so that they don't get damaged during cell division. So there are two reasons for the condensation step. Makes them easier to move, and it helps protect them from during that period of time that they're exposed to the cytoplasm. So at some point, as I've continued to say, we need to see within the cycle that cells move through a period of time when we're going to see the chromosomes being replicated. So we can actually think about the flow of the chromosomes during cell division in this diagram. It reinforces the idea of what happens to the chromosomes during cell division. So if you can make sense of what this is saying about what happens to the chromosomes, it will really help you understand what's going on in cell division. But it also reinforces the fact that we really do use the term chromosomes somewhat loosely. So this is a chromosome. We never actually see it in this form because remember, prior to cell division, it's in its chromatin form. It's not in its condensed form. But at some point prior to cell division, that chromosome is going to get duplicated. And so this, and I've never come across any biology professor that doesn't use this terminology. We call this a duplicated chromosome. Or replicated chromosome, because technically speaking, this whole thing is the chromosome composed of two sister chromatids. But if you just use the term chromosome, you really don't know whether you're talking about chromosome up here or, as you'll see, chromosome down here at the bottom. So most biology professors that I've ever come across, and our textbook authors do this as well, refer to this as a duplicated chromosome or a replicated chromosome to differentiate it from this single DNA molecule that is a chromosome. Then once the two sister chromatids separate, these now become daughter chromosomes. So once the sister chromatids separate, which they will do during the division process, we refer to them as daughter chromosomes. So again, we have a tendency to use the term chromosome somewhat loosely, but if you can remember the difference between chromosome, duplicated chromosome, and daughter chromosomes, it will help you so much in understanding what happens to the chromosomes during the two types of cell division that we need to talk about. So let's take a look at the cell cycle. So here is the cell cycle. It consists of two divisions or two periods of time. What is referred to as the mitotic phase, which is the time period during which cell division actually occurs, and it constitutes about 10% of the cycle. And then a period called interphase, which used to be called the resting phase, but we now know that it is anything but restful. The cell is extremely active. It's just that that activity is chemical and biochemical, so it's generally invisible under a microscope, but there's a lot going on in here. Now, you'll notice that there's a reference to G1 and G2 here, and the reason why, and it's not here on this, well, yes it is, but it's covered by this. For me, it's covered by this. So I'm going to move this out of the way for a minute. I'm not sure whether you see that. But I had to move something from the top of the slide so I could see the top. So interphase is now recognized as being broken up into three separate regions. S phase, which is the time period during which we know the amount of DNA is increasing. It's doubling. And so if the amount of DNA is doubling, that tells us that this is when the chromosomes are being copied. It's not a fixed period of time. It varies from cell type to cell type, but it is a recognized period of time within the interphase where we know definitively this is when the chromosomes are being copied. On either side of that, then, are two periods that have become known as gap 1 and gap 2. I hate the use of the term gap, and the reason why is because gap implies nothing's happening, and nothing could be further from the truth. Because there's a lot going on here. During the G1 period, the cell is actually recovering from the division process. It's freshly moving out of division. It's freshly formed. It needs to produce lots of protein. It needs to produce lots of lipid. It needs to produce lots of endoplasmic reticulum, Golgi body, mitochondria. If it's a plant cell, chloroplasts as well. It needs to do a lot, and most of that takes place during the G1 period of time. Does it extend on into S and G2? Oh yeah, sure. Don't get the idea that growth stops just because the cell is now in S and duplicating the chromosomes, or it's gone into G2. Growth is a constant part of the interphase. It's all about growth. So the cell is constantly increasing in size, increasing the amount of protein, lipids, sugars, everything is increasing during the interphase. But because of the identification of S, without being able to really see anything happening, just being able to measure the amount of DNA and recognizing that it's increasing, and then labeling the two periods on either side as gaps because you couldn't see anything. Well, that was just a function of using light microscopes to view things. But if we look at it in terms of the chemical processes that are going on, then there's a huge amount of chemical processing going on during the whole of interphase. When we get around to gap 2 or growth 2, as I prefer, there it's geared more towards the cell getting ready to divide again. Because if a cell gets through the G1 period and enters the S period, there is generally a commitment to dividing again. And so once you start S and you move on around to G2, basically what you find is the cell is gearing up to undergo division again, because there's this general commitment, if you enter S and move on towards G2, that there is a commitment to dividing again. And if we were to talk in detail about the regulation of cell division, we would talk about how that is accomplished. But suffice to say that once you get past G1, if a cell gets past G1 in the cell cycle and enters into S and moves on around towards G2, there is going to be a general commitment to dividing again. If it doesn't move past G1, then it moves into a stage that has become known as G0, and that's typically a maintenance mode, where the cells are not growing, they're not preparing to divide, they're just maintaining themselves. And it shouldn't come as much as a surprise for you to realize that the vast majority of the cells in your body are in that maintenance mode most of the time, particularly once you get to your adult stage. It's only when you need to do the repair and replace that the cells will actually actively divide again. During the time that you're growing and maturing and developing, well then, yeah, the cells will be growing and dividing on a regular basis. But once you reach your adult form, the vast majority of your cells, are not even in this cycle. They're sitting off to the side in what is referred to as G0 these days. Think of it as maintenance mode. So this is the cell cycle. What we're going to do now is take a look very specifically at the mitotic phase, which you can see is divided into two sections, mitosis and cytokinesis. And I want to stress something that is actually here, because this is a real issue that a lot of freshman biology students have. Confusing cytokinesis as being part of mitosis. Notice that mitosis here is strictly listed as division of the nucleus, which it truly is. That is what mitosis is all about. It's division of the nucleus and nothing else. Cytokinesis is division of the rest of the cell. So when the cell actually splits into two separate daughter cells, that's cytokinesis. But before that can happen, you have to separate the two copies of the chromosomes that you've created back here in the S phase of interphase. So there are three distinct steps in cell division. You need to make copies of the DNA. That takes place back here in S. You need to then separate those copies. That's what mitosis is all about. Starting with a single nucleus, and at the end of mitosis, you have two nuclei, each containing a complete set of identical chromosomes. So now you've created two separate copies of the chromosomes. Now you're ready to divide the cell, and that's where cytokinesis kicks in. The reason why it's easy to confuse cytokinesis as being part of mitosis is because cytokinesis invariably occurs in parallel with the last step of mitosis. So it's real easy to think of cytokinesis as actually being part of mitosis under those circumstances. Just be aware of that and make sure that you don't make that mistake, particularly with exam questions. It's so easy to do. It's really important to recognize that mitosis simply involves division of the nucleus, taking one nucleus and creating two identical nuclei by separating the sister chromatids. Once that is accomplished, then you can divide the cell into two new daughter cells, and that's accomplished by cytokinesis. Make sure that you bear that in mind. Hopefully you won't make that mistake. So we've talked a lot about these and what these involve. You can, I'm just going to put in here, just to reinforce again. Just to make sure that it's clear that these are growth periods. So this is just talking about, these slides are just basically covering everything that I've just talked about with the cell cycle. Diagram, again, the fact that cytokinesis is separate from mitosis. So to learn the stages of mitosis, here is an easy mnemonic. P at. Prophase, metaphase, anaphase, telophase. PMAT. So if you are struggling to try and remember the steps in order, just remember PMAT. Prophase, metaphase, anaphase, telophase. PMAT. And each of these phases is identified by what's going on with the chromosomes in the nucleus. Because remember, mitosis is all about division of the nucleus and separation of the sister chromatids, eventually. So we need to remember that that's what this is all about. That's what mitosis is all about. So we recognize each of the four stages, or phases as they're referred to here, by focusing on what goes on in the nucleus and what goes on with the chromosomes. So let's take a look at each of the steps in sequence. So here is a picture of an animal cell. It's in interphase. It's not begun dividing yet. You can see the microtubule structure. And these are important because it's going to give rise to the spindle. The microtubules are going to give rise to the spindle that's going to guide the movement of the chromosomes. So they're staged yellow or stained yellow. So right now they haven't organized themselves into anything of much. They're just a disorganized mass. Here's the nucleus. And again, you can't see the chromosomes in here. They're in their chromatin form, which they will be as long as the cell is not dividing. So once we move from interphase into the first stage of mitosis, prophase, the key thing here is that the chromosomes begin the condensation process and they start to become visible. So compare this nucleus with this nucleus. Clearly the chromosomes are becoming visible. They're condensing now. They're going through that coiling and supercoiling process to condense down to that chromosomal structure that we saw in the photo a few slides back. And remember, the chromosomes have already been duplicated, so it's the two sister chromatids that are condensing together, held together by the centromere. Can't see all of that yet because they haven't condensed into really defined chromosomal structures yet, but they're in the process of doing that. Because this is an animal cell, the spindle is going to form via what is referred to as centrosomes that contain centrioles. And again, if those sound a little bit fuzzy, you might want to go back and take a look at the chapter where we talked about the organelles in the cells. As I said, I believe that was chapter 5, because all of that was talked about in chapter 5. These are going to give rise to the spindle structure. So you can see the two centrioles here. They're going to move away from each other. So this one's going to move in this direction. And this one is going to move in this direction. And as they move away from each other, moving to opposite sides of the nucleus, they're going to create this spindle structure that's going to form a cage around the nucleus. Now, as the chromosomes finish their condensation and become distinctive chromosome structures, the spindle is going to want to engage with those chromosomes because the microtubules of the spindle are going to organize and move the chromosomes around. And there are structures in the centromere region, that region where the two sister chromatids are held together, are linked together, that will engage with the microtubules. Their technical name is kinetochore. We don't use that very much in this class, but they are referred to as kinetochores. And those are the structures that the microtubules attach to. And there's a kinetochore as part of the centromere region of each sister chromatid. And what's important here is that each of the chromatids is connected to one end of the spindle. Each chromatid being connected to one side while its partner is connected to the other side. So this one is connected over here, and this one is connected here. And you can see that that is true for all of the sister chromatids. They all have microtubules connecting to them, but one sister chromatid is connected to one end of the spindle, the other sister chromatid is connected to the opposite end of the spindle. And that should tell you something about what is going to happen when these sister chromatids separate. Obviously, the aim here is for each chromatid to move towards its particular end of the spindle and move away from each other. And given that they are identical to each other, that means that one identical copy of the chromosome goes to one end of the spindle, one identical copy of the chromosome goes to the other end of the spindle. Because remember, each chromatid is an identical copy of the original chromosome. So in order for the microtubules to engage with the chromosomes, the nuclear envelope needs to degrade. And so you can see that as part of the later stage, part of prophase. So this would be early prophase, and this would be late prophase. Now, if you are researching this outside of the textbook, maybe you're looking at the Khan Academy videos, or maybe you're looking at some other resources, you may see reference to something called prometaphase. Prometaphase has been developed by some cell biologists, the people who study this, as a way of dividing early and late prophase into two separate phases. And so if you think about the four phases of mitosis, prophase, metaphase, anaphase, telophase, prometaphase sits between prophase and metaphase. And it really constitutes what in this diagram I've labeled as late prophase. So just in case you come across that, that's where that comes from. But in this class, we only use prophase, metaphase, anaphase, telophase. So we're not going to worry about prometaphase. So in late prophase, the nuclear envelope breaks down so that the microtubules of the spindle can start to engage with the chromosomes and start moving them around. So what is that movement all about? Well, initially, it's to get all of the chromosomes excuse me. Not sure why I'm sneezing this morning, but I did. Anyway, the initial movement is to get all of the chromosomes lined up in the center of the spindle structure, what is referred to as the metaphase plate or metaphase equator. This is a virtual line across the center of the spindle. And that's what all of the chromosomes line up on this virtual line called the metaphase plate. And that's how you recognize metaphase. If you see the chromosomes more or less lined up in the center of the spindle, and here you can see a picture of an animal cell in metaphase, I know it doesn't seem like the chromosomes are necessarily lined up in the middle of the spindle, but they are. You have to use your imagination a little bit to recognize that. But this is a classic metaphase cell. The next step then is to separate the sister chromatids. Now that we've got them all neatly lined up in metaphase along the metaphase equator, we want to separate them. So the next stage, anaphase, is where the centromeres break So the centromeres break to allow the sister chromatids to separate from each other. One chromatid moves to one end of the spindle, the other chromatid moves to the other end of the spindle. And remember, each of these chromatids are identical to each other. So once they separate, they become identical copies of the original chromosome. So we've got one copy of the chromosome going this way and the other identical copy going this way. So now we've got two identical sets of chromosomes, each moving towards the opposite ends of the spindle. And they are going to form the complete set of chromosomes that are going to be incorporated into the nuclei of each of the two daughter cells that are going to be the product of this division. So here you can see the nucleus starting to form up again. The chromosomes have already started to decondense. The nuclear envelope is starting to reform. The spindle is breaking down. Basically, this is reversing everything that we saw going on in prophase. Oops. Helps if I spell this correctly. So reverses prophase. And you can see that even though technically speaking, this is telophase of mitosis, that the separation of the parent cell into two daughter cells has already started. That means cytokinesis has started. In parallel, with telophase. This is the reason why it is so easy to think that cytokinesis is actually part of mitosis, because it's occurring together with telophase, which is the last step of mitosis. So just make sure that you keep that separation, that cytokinesis is a separate step from mitosis. It is not part of mitosis. It's completely separate. Mitosis, again, division of the nucleus. Cytokinesis, division of the cell. Really important to keep those two separate. So now we have the 4 stages. How do we recognize prophase? Well, the chromosomes condense and become visible. The spindle starts to form. Later in prophase, the nuclear envelope breaks down and the spindle starts to engage with the chromosomes and moves them towards the center of the spindle. When they reach the center of the spindle, we've arrived at metaphase. That's characterized by the chromosomes lining up on the center of the spindle. Once the sister chromatids start to separate, when the centromeres break down, we have anaphase, the 2 sister chromatids, now daughter chromosomes, technically speaking, start to move to opposite ends of the spindle. Once they arrive there, a new nucleus starts to form around them, the nuclear envelope starts to reform, the chromosomes start to decondense and return back to the chromatin form again. So the supercoiling and coiling start to unravel and they return back to their chromatin form. The spindle breaks down, and because it can, cytokinesis, the actual division of the cytoplasm, begins, even though, remember, technically it's not part of mitosis. So that's mitosis and cytokinesis together. So here's the BioFlix animation, and again, this is available in the resources section. And it's about a 3.5-minute animation that walks you through the stages of mitosis. And I've forgotten right now whether they refer to prometaphase. I believe they refer to prometaphase in the animation. So if you take a look at this, you may see reference to prometaphase. Remember again, prometaphase is not a phase that we are recognizing in this class. In the animation, if they're using prometaphase, think of that as what I've referred to as late prophase, and what they're referring to as prophase, I've referred to as early prophase. I'm sorry that there's this little bit of confusion. I can only use the resources that are available, and unfortunately, this animation was developed more for the advanced biology class, the general biology 1 class, and there they actually do have the prometaphase stage in the textbook, so unfortunately the animation reflects that. This is the only BioFlix animation for mitosis, so we have to live with that, even though the textbook doesn't refer to it. But hopefully you get the idea. So, given that cytokinesis is something that has to take place in order for the final division of the cell to take place after the 2 nuclei have formed up, we can't just stop at that point, because it turns out that because of the differences between animal cells and plant cells, cytokinesis is necessarily different between the 2. And you might ask, well, what do you mean? Why should it necessarily be different? Well, what is the big difference between an animal cell and a plant cell? Hopefully you remember that plant cells have a cell wall, and they can't function without an intact cell wall. So while cytokinesis in animal cells is really easy, the cells just simply split into 2 via this cleavage furrow, which is triggered by actin protein, which makes up microfilaments. Is kind of like a piece of elastic shrinking down around the cells until it literally pinches the cell in half. Animal cells can do that. They don't have to worry about a cell wall because they don't have one. but plant cells do have a cell wall. And so in order for the plant cell to survive the division process, you need to form the cell wall as part of cytokinesis. So cytokinesis is actually carried out by forming the new cell wall that's going to actually divide the two daughter cells or separate the two daughter cells from each other. It begins by a series of vesicles that line up between the two nuclei in that region where the metaphase plate was located, right? Where the chromosomes lined up during metaphase. It's along that line that these vesicles line up. They eventually increase in size to the point where they link up and form this thing called a cell plate. And remember, this is expanding not only up and down, but also out of the diagram and back into the diagram. It expands in 3 dimensions. And eventually it merges with the existing cell wall that surrounded the original parent cell. And so in the process of forming this new cell wall via the vesicles and the cell plate, you accomplish cytokinesis. You split the parental cell into 2 daughter cells, but at no time is there an incomplete cell wall surrounding this whole original cell or the 2 resulting daughter cells. So it's the presence of the cell wall and the importance of maintaining the integrity of that cell wall around the cell during the cytokinesis process that means that cytokinesis necessarily has to be very different in plant cells versus animal cells. So that's also something to remember. So that's the basis of basis of mitosis. The 4 steps of mitosis: PMAT, prophase, metaphase, anaphase, telophase, and how you go about identifying each of them. And then cytokinesis is the division of the cytoplasm, the original parental cell, into 2 daughter cells, and the differences between animal cell cytokinesis and plant cell cytokinesis. Now, as I mentioned earlier, cell division is tightly regulated, and there's a really good reason for this. We don't want cell division to occur arbitrarily. Think about when you cut yourself. Do you want cell division to begin when you cut yourself? Oh yeah, yes, you most certainly do, because you want to seal up that cut and reform the skin as the barrier against pathogens. But once that barrier is healed, do you want cell division to continue? And obviously the answer is absolutely not. So there needs to be a way of telling the cells, yes, it's okay to divide, or no, it's not okay to divide. And so there are protein signals, both internal to the cell and external to the cell, that tell the cell that it's okay to divide and not okay to divide. So generally, the signals on the inside of the cell tell the cell that it's okay to pass from G1 through S onto G2 and then into mitosis and on again around the cell cycle. And so long as those internal signals are present, the cell will continue to move around the cell cycle, growing and dividing and producing more cells. The external signals are what tell the cell whether to stay in the cell cycle or to exit the cell cycle into that maintenance phase, the G0 stage. And so it's the combination of these 2 sets of signaling molecules, what we refer to as growth factors. So I'll put in here, AKA growth. That regulate the ability of cells to divide or not to divide. And these are very, very powerful molecules. Experiments have been done that demonstrate that the simple exposure of a cell to another cell in a different stage of mitosis will trigger the first cell to immediately move to well, I shouldn't say mitosis, a different stage of the cell cycle, will immediately trigger the first cell to move to the stage of the cell cycle that the second stage is in, or the second cell is in. These molecules are really, really powerful. they can override the normal programming of the cell and force it to move to a stage in the cell cycle that it was probably not going to get to for some time. And in so doing, it skips over the intervening stages. So the one that really is the gee whiz one for me is to take a cell that is in G1. Okay, so it's just finished dividing. It's not going to divide any time in the near future. 90% of the cell cycle is still in front of it before it would even begin to divide again. When you merge that, when you fuse that to a cell that's already undergoing mitosis, the cell in G1 immediately starts mitosis. It doesn't even bother to duplicate its chromosomes. It doesn't even bother to hesitate. It doesn't go finish G1. It doesn't pass through S. It doesn't pass through G2. It immediately starts forming a spindle and starts condensing the chromosomes. It enters mitosis, even though it's not really set up for it. That's how powerful these growth factors are. They're really, really powerful. So under normal circumstances, they wield a very powerful control over the ability of cells to grow and divide. And they stop the arbitrary division of cells and regulate when cells are given permission to divide and when they're not given permission to divide. What cancer constitutes is when that control system goes haywire. So there are two basic control mechanisms that all cells recognize that allows them to divide or not divide. One is that they need to be attached to a surface. That's called anchorage dependence. So this would involve cells being attached to each other via their extracellular matrix so that they recognize that they are attached to something. The one exception is blood cells. So cancer of the blood can involve cells that can divide even though they're not anchored to something. So the leukemias. But most other cells, even if they're cancer cells, they've got to be anchored to something in order to be able to divide. The second one is called density-dependent inhibition. And this we can demonstrate in the laboratory very easily, that if there's no room for the cells to divide, it doesn't matter whether they're anchored to something and probably would divide. If there's no space for them to divide, they simply won't divide. But this is where cancer cells become a real issue, because cancer cells very specifically do not obey density-dependent inhibition. And this is part of the reason why cancers, and particularly what are referred to as malignant tumors, which is really what a cancer is. As you'll see, there are two different types of tumor, only one of which is technically cancer. But when the cells start growing in places where they're not supposed to grow, which is the definition of a malignant tumor or cancer, they're growing in a location that is not their point of origin. This is when they become deadly, because they are now going to occupy space that would normally be occupied by healthy cells. And they're going to put pressure on healthy tissue. They're going to take resources away from healthy tissue. They're not going to play nice with the other cells. They're going to push them out of the way. They're not going to obey density-dependent inhibition. And this is typically when a cancer becomes deadly, because the cells are now occupying space that really should be occupied by healthy tissue. They should not be there, but they're not obeying the density-dependent inhibition signaling. And that's basically what's wrong with cancer cells. They do not obey these signals that tell them don't divide. And the reason why is because the growth factor proteins are produced by genes that normally regulate the ability of cells to grow and divide. That's what these proteins do. They are product of genes that are involved in, and the protein products that they produce are involved in telling cells to grow and divide. And they're typically switchable. They can be in an on position or an off position. But if the genes get altered, mutated, then these proteins change their function. And the big deal change in function here is that rather than being switchable, they get locked in a permanent position. And unfortunately, the permanent position is permanently on. In other words, they're sending the signal to the cells, it's okay to divide. And so this is typically what happens. These genes, unfortunately, have been named oncogenes. Which at first glance suggests that these are cancer-causing genes, which is an unfortunate state of affairs, because really there is no gene present in your chromosomes whose job it is to make you sick. All of these genes have normal functions, including these genes that have been labeled as oncogenes. They have a normal function. What's their normal function? To tell the cells whether it's okay to divide or not, to regulate cell division. The problem is that when these genes get mutated, they change their role. Rather than regulating cell division, now their role is to tell the genes to divide. They've taken on an abnormal role. So the one thing that we know in all of this is that in order for cells to change from normal healthy cells to pre-tumor cells to tumor cells to precancer cells to cancer cells involves multiple gene changes. So it's not just a single gene that needs to be altered. It's usually multiple genes that need to be altered. And we typically think that about 5 genes and 5 key genes need to be altered for a cancer to develop. So I'll put a note in here. Let's get this going here. So just to add that in, it requires multiple changes to multiple genes in order for a cancer to develop. So there, as I mentioned just a minute ago, there are two different types of tumors. There's what is referred to as the benign tumor. This is a tumor that's growing at the point of origin, and it's referred to as benign because these are typically not deadly. And you can generally live for an extended period of time with a benign tumor. The problem becomes that most benign tumors do not remain benign indefinitely. They can become malignant. And malignant means that cells from the original tumor break free from that original tumor, migrate to another location in the body, and start growing there. The process is called metastasis, and you can see the name down here, metastasis. We say that the cancer has metastasized. And so when these cancer cells move to this new location and form a new tumor and start growing, that's when it becomes malignant, and that's when we say you have cancer. So whenever you have a benign tumor, you don't have cancer, but that is a precancerous condition. If cells from that benign tumor metastasize, move to another location and start a new tumor in a different location other than its point of origin, now you have a malignant tumor, and now you officially are diagnosed with cancer. And unfortunately, for some types of cancer, you only really recognize that it has occurred because of the metastases. It is really important, however, to pay attention to what our body is telling us, because many times, if you know the signals, you can recognize that there are problems. And one of the things that we know for certain is that the earlier you recognize the presence of the cancer, particularly while it's still in its benign form, the greater is the chance of a successful cure. Because if you're dealing with a benign tumor, you're just dealing with a single tumor, and if it can be removed surgically, then no harm, no foul. You take it out and that's it, that's the end of it. If it has potentially metastasized, and there are ways of figuring that out, then now you're dealing with the problem of not only the benign tumor, but the possibility that there are cancer cells now loose elsewhere in your body and you have no clue where they are. So now you need to start thinking in terms of chemotherapy and radiation therapy as a systemic treatment, meaning you're not treating a local region, you've got to treat the whole body, the whole system. That's what we mean by a systemic treatment, because you just don't know where the cancer cells are anymore. So you can't take the chance that there are free cancer cells floating around in your body, because it only takes a few to start a tumor. So you've got to treat the whole body systemically with chemotherapy and radiation therapy. And while I've been fortunate enough not to require chemotherapy and radiation therapy, I have had some skin cancers that have had to be treated, but they were benign. I do have friends and colleagues that have had to have chemotherapy and radiation therapy, and based on what they've told me, it is not fun. There is nothing pleasant about chemotherapy and radiation therapy. It sucks, and it sucks big time. So obviously the moral of the story here is the earlier that you can identify the presence of the tumor, preferably while it's benign, the easier it is to treat it, and probably the less painful that treatment is going to be. So what is the basis for metastases? Well, the problem is that we have two pathways for the cancer cells to move around. The bloodstream, which you already are aware of, and another system called the lymph system, which captures all the fluid that leaks out of the bloodstream. So what is referred to as interstitial fluid, the fluid that surrounds the cells, that's the clear fluid that leaks out of a cut after the blood stops flowing. That's actually blood plasma, by the way. It leaks out of the bloodstream. All of that is captured in the lymph system, and returned back to the bloodstream just before the blood enters the heart to keep the volume up and allow it to be pumped freely around the body. Periodically along the lymph system are lymph nodes. These are part of the immune system, and in there are cells that recognize how to recognize pathogens and remember how to make antibodies against them. Because any pathogen, any cancer cell, any virus particle, any bacterium that might be loose in your body is almost certainly going to show up in the fluid that's captured in the lymph system. And so it makes sense to have monitors monitoring the fluid to see if there are any pathogens on the loose and to start making the antibodies necessary to get rid of them. And cancer cells are, under most circumstances, recognized by the immune system and nailed before they become a problem. That's what part of the immune system is to do. It is to monitor for the presence of cells that are becoming cancerous or precancerous or pre-tumorous, and to kill them before they can do any damage. Likewise, there are a series of genes called tumor suppressor genes, the most famous one being one called P56, the P56 tumor suppressor gene, that very typically gets altered as part of many cells becoming pre-tumorous and precancerous, because this particular gene and the tumor suppressor genes in general are supposed to trigger the cells to die, a process called apoptosis, if they become risky cells. So cells that have genetic damage, chromosomal damage, other types of damage that make them risky in terms of allowing them to divide. The P56 tumor suppressor gene and other tumor suppressor genes are supposed to trigger apoptosis and cause the cells to die. It's type of a programmed cell death or cell suicide. Better that those few cells die than they become a problem for the rest of the body and create a situation where the rest of the body could very well die. So these genes are ones that very typically get altered as cells become pre-tumorous, tumorous, precancerous, and cancerous. So it's not just the growth factor genes, the external signaling genes or the internal signaling genes, but it's also these tumor suppressor genes that get altered. You can see that there's this whole series of checks on cells that are supposed to prevent cells becoming pre-tumorous, tumorous, precancerous, and cancerous. And by and large, the system works. You have to be really unlucky to develop cancer. But yet it still happens. And we know it happens with relative frequency and with apparently increasing frequency. Now, some of that may simply be because we live longer now, because we know that cancer is typically a condition of old age. The longer you live, the more likely it is that you will develop some form of cancer. In fact, as far as prostate cancer is concerned in men, if you live long enough as a man, you will develop prostate cancer. That seems to be the situation based on looking at prostates of men at various ages after they die. The longer they live, the more likely it is that the prostate cancer will have begun. If you are young enough when it begins, then they treat you for it because you will live long enough that the cancer will become an issue. But fortunately, prostate cancer is a very slowly developing cancer. So if you are relatively old, they probably won't do anything by virtue of the fact that you will die of old age before the prostate cancer could potentially kill you. It's just one of those things. Then, of course, we do know that there are some genes that are identified as being associated with different types of cancer. So there is what is known as the BRCA complex, otherwise known as the BRCA complex or BRCA complex, depending on how you're taught to pronounce it. So there's BRCA1, BRCA2, and BRCA2A. They're all associated with breast cancer, ovarian cancer, cervical cancer, and it's now been shown to have a connection to some forms of testicular cancer. But what most people don't tell you is that those genes are only associated with 15% of 15% of breast cancer. So if you are screened for the presence of those genes and you don't have it, well, that's good news because you won't get breast cancer from those genes, but it doesn't mean that you can't get breast cancer. And while we're talking about breast cancer, gentlemen, you need to be aware of the fact that you also have breast tissue and you can develop breast cancer. And it is typically more deadly in males than it is in females because women are taught from a very young age to do breast self-exams and to pay attention to any changes because it's very common in women. It's not as common in men, but because men tend not to test or to check their breasts on a regular basis, by the time they recognize that the breast cancer has developed, it's in an advanced stage, and that makes it really difficult to treat. So the moral, the moral of the story is to pay attention to what your body's telling you. Take advantage of the fact that your body can signal to you that things are not as they should be, and pay attention to those signs. And make sure that you're doing the things that you know that can at least prevent the likelihood of the development of cancer. Now, unfortunately, there is also an inherited component. We do know that some types of cancers show an inherited component, meaning they pass from one generation of a family to the next generation. And so we also need to recognize that if you have a close relative, meaning brother or sister, that develops a particular type of cancer, that increases your risk substantially because you are drawing from the same set of chromosomes and genes. You have the same parents. If it is a parent, then there is an increase in risk, but the increase is not as great as if it was a brother or sister because you only share about 50% of the same genes with a particular parent. If it is a relative of your parents, an aunt or an uncle, then the risk is even less. If it is a grandparent, the risk is substantially less as well. But there is always an increased risk over the background risk of getting a particular type of cancer if you have a relative. And the more closely related they are to you, the more increased is your risk potential. But there are some things, even if you have a family history of particular types of cancer, that you can do to decrease the chances, because remember, we're not just the product of our genes, something that we will reinforce in the next chapter. So not smoking, not lying out in the sun, there is no such thing as a healthy tan. The fact that your skin is tanning is the indication that it's already been damaged. That's your skin's response to the sun damage. So to say that a tan is healthy is an oxymoron, because it is anything but, because you've damaged your skin in order to get it to tan. And that damage is in part damage to the DNA, or potentially damage to the DNA, which could lead to skin cancer, which is one of the more dangerous types of cancer. There are all sorts of lifestyle things that you can adopt that will reduce your chance of developing cancer. And most of us know what most of those sorts of things are. A healthy diet, not smoking, controlling how much we drink and not abusing alcohol, not lying out in the sun, not using a tanning bed, all of those sorts of things can help us prevent the development of cancer. Is it 100%? No, of course not. No one can guarantee that you'll never develop cancer. But there, as I said, there are some lifestyle things that you can do that will limit your chances. So we talked a little bit about the types of treatment. Surgery is the preferred way, because if it's benign, you can cut it out and that's it, no further treatment required. If there's the potential for metastases, then we need to take a look at the use of radiation therapy or chemotherapy or a combination of the two to systemically treat the whole body, to target the cells wherever they may be. So radiation therapy can be a local therapy, but it can also be a systemic therapy, depending on the nature of the type of cancer. And as I've said, these are the sorts of things that you can do to prevent cancer. So another timeout, another break, and we will. I will be stopping the recording at this point again. So if you want to take a break, by all means, feel free to go do so. Here are the concept check questions that I'll answer when we come back from the break. What are the two broadest divisions in the cell cycle? What are the two processes that are involved in the duplication of cells? What are the differences between a benign tumor and a malignant tumor? And there is a really cool looking organism called a plasmodial slime mold. These are very real, that consists of a large cytoplasmic mass, a truly large cell with multiple nuclei. Based on what you know about mitosis, how can you account for the existence of this type of organism? I'll see you on the other side of the break.